We usually think of death as a failure, right? An ending. But what if I told you that inside your own body, death isn't just an end? It's this incredibly precise, beautifully choreographed process that literally keeps you alive and healthy. Today, we're going to dive into the absolutely fascinating world of cellular suicide.
It sounds like a total paradox, doesn't it? But really, this question is at the very heart of our biology. It turns out the ability of our cells to die on command is every bit as important as their ability to grow and divide. And this is not some rare, once-in-a-while event. Just look at this number, 10 billion. That's the estimated number of cells that die in your body every single day on purpose. It's this constant, massive, internal housekeeping effort that maintains the delicate balance that we call life.
So what is this process called? Well, the official term is apoptosis. And the keyword here, the one you really need to remember, is programmed. This isn't some chaotic accident or injury. It's a pre-installed self-destruct sequence, hardwired into the genetic code of almost every single one of our cells.
And you can see it at work all the time, in some pretty critical ways. Apoptosis is actually the sculptor that carved out the spaces between your fingers and toes when you were just an embryo. It's the process that gets rid of cells infected with a virus, it destroys cells that have dangerous, cancerous mutations, and it even weeds out immune cells that might otherwise turn on us and attack our own bodies. I mean, without it, we just couldn't function, period.
Okay, so that leads to the next big question. How does a cell actually get the memo that its time is up? Well, as you can imagine, there are some pretty sophisticated signaling systems in place to make this ultimate life or death decision.
Basically, apoptosis gets triggered down two main highways. First, you have the extrinsic pathway. You can think of this like getting a tap on the shoulder from another cell. It's an external signal that basically says, hey, you're no longer needed. Then you have the intrinsic pathway. This one's an inside job. It gets triggered by internal stress signals, like, say, if the cell's DNA is damaged beyond repair.
Now, no matter which path gets taken, they both lead to the exact same destination, activating this family of proteins called caspases. You can think of them as a cellular demolition crew. First, the initiator caspases get the signal. Then, they turn around and activate the executioner caspases. And once those executioners are switched on, believe me, there is no going back.
And the demolition itself is incredibly clean and organized. The cell shrinks down, its DNA gets neatly chopped up into little pieces, and the outer membrane bubbles off into these tidy little packages called apoptotic bodies, which your other cells then come and clean up. And maybe the most important part? This all happens without causing any inflammation. Unlike other messy forms of cell death that can trigger a big immune response, apoptosis is silent, efficient, and clean.
But, of course, a decision this final needs some super strict regulation. I mean, you can't just have cells self-destructing for no good reason. So who are the gatekeepers? Who makes this ultimate call?
Well, the answer lies with a super important family of proteins called BCL2. You can think of these proteins as the master regulators, the ones that are constantly weighing all the signals for life against the signals for death inside the cell.
Really, the best way to picture this is as a molecular tug-of-war. On one side of the rope, you have the anti-apoptotic proteins, team staying alive, and they're fighting to keep the cell going. On the other side, you have the pro-apoptotic proteins, team time to go, pushing for self-destruction. The entire fate of the cell is literally hanging on the outcome of this internal conflict. And this is the tipping point. If team time to go wins that tug-of-war, they essentially poke holes in the mitochondria, the cell's power plants. This releases a molecule called cytochrome C, and that is the ultimate irreversible signal that commits the cell to apoptosis. It is the point of no return.
So, as you can see, this balance is incredibly delicate, and when this perfectly tuned system gets thrown out of whack, well, the consequences can be absolutely devastating.
And the stakes really couldn't be higher. If you have too little apoptosis, cells that are supposed to die don't. This can lead to cancer, where cells basically become immortal, or autoimmune diseases, where those self-attacking immune cells just never get eliminated. On the flip side, if you have too much apoptosis, we start losing essential cells we need. This is a huge factor in neurodegenerative disorders like Alzheimer's, and it's also what causes a lot of the damage after a heart attack.
And that's exactly why understanding apoptosis isn't just some abstract academic exercise. It has become a central focus in modern medicine and therapeutic research. It's truly key to tackling some of our biggest health challenges. So, if we understand the system this well, can we actually control it? That is the billion-dollar question, right? And the answer is leading to a complete revolution in how we design new therapies.
We can already see this happening with new kinds of treatments. For cancer, scientists are developing BCL2 inhibitors. These are drugs that essentially go in and force those immortal cancer cells to finally hit their self-destruct button. For things like neurodegenerative diseases, the goal is the complete opposite, using Caspase inhibitors to block the death signal and protect precious neurons. It's all about trying to restore that critical balance.
So by learning the art of dying well at the cellular level, we are opening up entirely new frontiers in medicine. This research gives us a toolkit to potentially intervene in countless diseases, which leaves us with a really powerful question for the future. If we can truly become the masters of cellular life and death, what could we conquer next?